Hi everyone, this is the introduction to the case study for variant discovery and genotyping. This will be presented by several members of the bioinformatics core team, which is part of the Harvard T.H. Chan School of Public Health. So we hear a lot about personalized medicine, precision medicine, genomic medicine, or other descriptors of the same thing. What this basically means is taking the genome of an individual or a group, also known as a cohort, with a disease and trying to determine how their genome differs from a healthy control or the known reference. Current technology does not allow us yet to sequence someone's DNA in one go. So we end up breaking it into millions of little fragments of around 100 characters or 100 bases. And then we compare them to a reference genome to best figure out where they could have come from in a process called sequence alignment. Once we have the location of each sequence fragment or read, we can then try to find where they differ from the reference or the healthy control, if you will. We check for individual single base differences or substitutions or larger insertions and deletions, with the focus on those differences that have an impact on so-called coding regions. That is, the regions that will be translated into proteins, our cellular machinery. So what is the scope of this case study? In this case study, we will focus on calling variants, SNVs or single nucleotide variants, and indels or small insertions and deletions. We will be doing this in germline samples, using command line tools and a best practice workflow that you will be able to reuse for your own projects. We will not be going over large structural variants. The workflow we will be following for this case study starts with sequence data, checking the quality of the sequence data, aligning the sequence reads to a known reference genome, followed by calling variants based on the read alignments. This will be followed by filtering and annotating the variants. And finally, we will query a set of known databases.